Imagine a time long before humans, before dinosaurs, even before most land animals roamed the earth. Our planet was a water world, teeming with strange and wonderful creatures. Among them were ancient fish, unlike any you see today. These fish were often covered in tough, bony armor. Scientists are now discovering that parts of this external skeleton held a secret. These bony plates, scattered across their bodies, were not just shields. Some of them were the very first versions of what we now know as teeth, but they weren't in the mouth. They were on the outside, part of their skin. Recent discoveries using powerful scanning technologies have peered deep inside ancient fish fossils. These scans reveal intricate networks of nerves and blood vessels within these denticles. This structure is remarkably similar to what we see in sensory organs. It suggests that these external teeth weren't primarily for biting or chewing food in the way we understand it. Instead, they might have helped these ancient fish feel their way through murky waters. They could have detected vibrations, changes in water pressure, or even the subtle movements of prey hiding nearby. Think about the scales of a shark. They are a modern example of these dermal denticles. Each tiny scale is structurally very similar to a tooth. This connection has been known for some time, but the sensory aspect adds a new layer of understanding. Let us journey back to the Devonian period, often called the Age of Fishes. This era, roughly 419 to 359 million years ago, saw oceans bustling with life. Among the most dominant and fascinating creatures were the placoderms. These weren't your typical goldfish. Placoderms were a diverse group of armored prehistoric fish. Their name literally means plate-skinned, a fitting description for creatures whose head and thorax were covered by articulated bony plates. This armor provided significant protection from predators, turning them into veritable underwater tanks. Some, like the fearsome Dunkleosteus, grew to enormous sizes, reaching lengths of up to 6 meters or more. They were apex predators, equipped not only with strong body armor but also with powerful, blade-like jaw structures made of sharpened bone, rather than true teeth like later vertebrates. These jaw plates could slice through the tough defenses of other armored fish and shelled creatures, making them formidable hunters in their primeval world. The dermal denticles, those tiny tooth-like structures embedded in their skin, played a vital role. These skin teeth could have helped them navigate, find food, or even detect approaching danger long before it was visible, providing a crucial edge in the struggle for existence. The development of jaws, a key feature in placoderms, was a major step in vertebrate evolution. For centuries, fossils have been our primary windows into the deep past, offering clues about the creatures that came before us. However, recent advancements in scanning technology have revolutionized paleontology. Techniques like synchrotron X-ray microtomography allow researchers to peer inside fossils without damaging them. They can create incredibly detailed three-dimensional reconstructions of internal structures, revealing features previously hidden from view. This technology is now unlocking secrets about the very first teeth. Applying these high-resolution scanning methods to the dermal denticles of ancient fish, particularly placoderms, has yielded astonishing results. Researchers were able to visualize the intricate internal networks within these tiny, tooth-like structures on the skin. What they found were not just simple, solid bumps. Instead, they discovered complex systems of canals and chambers. These spaces in life would have housed nerves and blood vessels. The presence of such extensive innervation strongly suggests a sensory function, a way for the fish to gather information from its surroundings. Section 4. The Great Migration: How Teeth Moved from Skin to Mouth. The evolutionary journey of teeth from the external skin to the inside of the mouth is a remarkable tale of adaptation and repurposing. If teeth originated as sensory dermal denticles on the outer body of ancient fish, how did they end up lining our jaws? The process was likely gradual, driven by the pressures of natural selection over millions of years. The key seems to lie in the close developmental relationship between skin and the lining of the mouth. The tissues that form our skin and the tissues that form the inner lining of our oral cavity share a common embryonic origin. As early vertebrates evolved, jaws became increasingly important for feeding. Initially, the edges of these primitive jaws might have been lined with the same kind of bony plates or denticles found on the rest of the body's armor. Perhaps denticles located near the mouth opening, already equipped with sensory nerves, began to assist in grasping prey or manipulating food items. Over time, any variation that made these oral-adjacent denticles slightly larger, stronger, or better anchored would have provided a feeding advantage. The sensory function likely didn't disappear immediately. Early intraoral teeth might have retained some sensory capabilities, helping the animal to taste or feel the texture of its food, or to precisely position prey within its jaws. However, as the mechanical demands of biting and chewing became more pronounced, the primary role of these structures would have shifted. Section 5. Rewriting the textbooks, a new chapter in dental evolution. This groundbreaking discovery about the sensory origins of teeth fundamentally changes our understanding of dental evolution. For a long time, the prevailing theories focused primarily on the mechanical aspects of teeth, their role in grasping, tearing, and grinding food. While these functions are undeniably crucial, the new evidence suggests that the story of teeth began with a much more subtle, sensory purpose. It implies that the genetic and developmental pathways for creating tooth-like structures evolved first to help ancient fish perceive their environment, long before these structures were co-opted for feeding within the mouth. The traditional view often posited that teeth evolved directly in the mouth in response to dietary needs. However, the outside-in hypothesis, supported by the sensory nature of dermal denticles, provides a more comprehensive and evolutionarily coherent narrative. It elegantly connects the scales of sharks, the bony armor of extinct placoderms, and the teeth of all jawed vertebrates, including humans, into a single evolutionary continuum. This unified view helps to explain why teeth and skin appendages like scales share such striking similarities in their development and structure. They are, in essence, variations on an ancient theme, a theme that began with feeling the world rather than biting it. This new perspective also helps to solve some long-standing puzzles in vertebrate paleontology. The nerves in our own teeth, which cause us so much pain during a toothache, are a direct legacy of this ancient sensory origin. Section 6. Ancient blueprints, modern wonders, what old teeth teach us today. The revelation that teeth originated as sensory organs on the skin of ancient fish isn't just a fascinating piece of evolutionary trivia, it has profound implications for modern science and medicine. Understanding the deep genetic and developmental pathways that govern tooth formation can provide valuable insights for fields like regenerative dentistry and bioengineering. If we can unravel the ancient genetic toolkit that first allowed skin to produce these complex mineralized structures, we might one day be able to coax our own tissues into repairing or even regrowing teeth. This ancient blueprint holds clues that could revolutionize how we approach dental health and tissue regeneration. In genetics, this discovery highlights the remarkable conservatism of evolution. The same fundamental genes involved in forming dermal denticles in a 400 million-year-old fish are still active in shaping our own teeth, skin, hair, and feathers today. Studying these genes in diverse living organisms from sharks to humans can help us understand their core functions and how they've been modified over time to produce such a wide array of structures. This comparative approach can identify key regulatory elements that could be targets for therapeutic interventions, potentially addressing genetic disorders that affect teeth and other ectodermal appendages, those structures arising from the outermost embryonic cell layer. Bioengineers, too, can draw inspiration from these ancient natural designs. The way these early sensory denticles integrated with nervous and vascular systems offers a model for creating advanced, biocompatible materials or sensory prosthetics. Imagine developing artificial skin grafts that not only provide protection, but also incorporate sensory feedback, mimicking the sophisticated capabilities of these primordial structures.